My name is Jessie Kissinger. I'm one of 3 joint PIs for the VEuPath project, together with David and Marianne McDowell. And today I'm going to talk about the redesigned homepage, and I'm going to spend a good bit of time letting you know the depth of the secrets that are sort of hidden on this page. And I wanted to take a minute to say that in the process of merging with VectorBase and VEuPath, we decided to take the best elements of each BRC and tools that had been developed, and then also use the opportunity to address some issues on the old sites that had been brought to our attention based on community feedback and fix them. And so if you were familiar with our old sites, and I know that some of you are, you might be looking for, you might be familiar with a page that looks something like this, that had sort of 3 main boxes in the middle and data and a bunch of tabs across the top. The new merged base, if you work on for pathogens and vectors now in VEuPath, will look like this. And if you haven't been to our sites before, this new site was specifically designed in part with you in mind. And that is, we wanted to make it easier for someone to come to our sites and be able to identify the tools right away and be able to answer their question right away in the absence of, extensive training or having to look up a fair amount. And I also wanted to spend some time going through today, and the reason you're at the workshop is that this is not just a website that you browse. It's a website where you can actually conduct an in silico experiment. And I think the meaning of that will become clear as we go through the course of the day. And even though we're going to teach you a lot today, there is so much more that can be done. Okay, so the homepage has 7 sections. And the first one here being the header. And there's a lot packed into this header. And the first thing I want to show you about the header is a secret property that it has. But anytime you click on this logo up in the corner, you will be taken back to this homepage, all right? And it doesn't matter what site you're on. This site happens to encompass all of the organisms within our resource now, which is over 400. But you could also be looking at Plasmo, right, or Tri-Trip. The same tools, the same layout, everything applies, okay? So what I'm telling you today will be relevant to whatever organism you happen to work on. All right, the release, we release bi-monthly, so you will be able to find exactly what version we're working on. And our newest feature, which is something that wasn't present on the Protis sites before, is a site search. And it does just what you expect a site search to do. You type in any sort of keyword, thing that you're looking for, and immediately, we have an exercise on it, so I'm not going to do it right now because it's the next thing you're going to do, will take you, and it can be a gene name, a protein product, a word publication, an author who might have submitted a data set. You can have a lot of fun playing with this. Underneath it are what are tabs, effectively, where you can have strategies, searches, tools, a workspace, data. It's a lot here about the data that's in our sites, about us, about the community, about publications, about submitting data, about usage and citation, who we are, some technical information, help, and contacting us. The contact us button, super important, all right? Because this is the main way that you can communicate with us, and you can communicate with us about anything, okay? And that's really important. Let me take this one back. I'm going to click on the magic button. There we are. Okay. In addition to in the header, you're going to find also our presence on social media. So you can go to our Twitter page, our Facebook page, and really importantly, I want to go to the YouTube channel page that I've already just gone ahead and bookmarked. And that is workshops like this. Right now it's being recorded. Tutorials prepared by the outreach team are listed here. And I think, as you can see, that in the just VEuPath webinars, there are 26 different recordings, tutorials. There are 30 for these related databases that David mentioned, ClinEpi, just introductions. How do you use things? How-tos. Some of these are from our older sites. You can tell by the UI. But some of the functionality, well, a lot of the functionality will still be there once you realize the differences between the layouts. And so there's a tremendous amount packed in here, and this is definitely a great way to hook up and find out about papers, to follow papers. Our outreach team is always tweeting and highlighting research, bringing it to our users' attention. The next part of the page is called the banner. And it's right here shown in light blue. And this is where we provide you, the user, with important messages, okay? In this case, we've let you know that we've merged and that we have released a new site. And you might go, you know, I just want to find this. I don't have time to learn the new site. You know, if you're trying to get to the older sites, right here, you would click on it, legacy sites, and you would be taken to the legacy PlasmoDB, TriTrip, Pyroplasma, whichever site you're interested in, as well as VEuPathDB. So, and we will also, when we advertise workshops, when we're going to be at meetings, there's a lot of information that appears in the banner. And the banner doesn't always appear on every page. So if you bookmark an internal page, please be sure to have a look at the front page from time to time. It's also the place where you can send us feedback about these new sites, all right? So if there's something that's really working or something that was confusing, please, we're still in beta. This is a chance for us to make updates to the sites. We'd like to hear from you. The right-hand panel, it's our third part of the website, you will find news releases. Because this is the ViewPath site, sort of the umbrella that contains all of our project sites, vector-based, Plasmo, Fungi, etc. You'll find all of the news in here. You'll have release notes on the data that have been released. Sometimes there will be other types of news. So there's going to be a forthcoming microsatellite variation data coming from the Powell lab. So there'll be a lot of news in here, and you can follow that. And our tweets, following the Twitter page, and new papers, research coming out, etc. It's heavily used, and so you can expand and close this that way, just by opening it and closing it. The center of the page is focused on if this is your first time using a ViewPathDB resource, or it's the first time that you're trying to do something new. Maybe in the past you just were looking at your gene, but now you need to know something about RNA-seq data or about phenotype data. You can take a tour of the site. You can learn how to get started. You say maybe you're interested in phenotypic data, and there'll be a description of some of the tools that are available. And you'll see there's images of phenotypes, etc. And the pages are explained in detail. And in the middle of this, if I just here read less, below that, again, are links to tutorials and exercises like you'll go through today that are very self-guided, self-paced to help you learn to do a particular new tool if you should want to do it. All right. So those are our 5 parts of the page now. So over here, we have searches also. And unlike a site search that is a catalog of every time a word or a term appears and where it appears in the site, these are strategic mining tools, right? And so maybe you came here with the notion of, I just need to find signal peptides. And I really encourage you, like, where would I find signal peptides of all these buttons? Where would it be? I encourage everyone to just start typing in this box. I got as far as Cigna, and here it is, predicted signal peptides. It takes me right there, and I can remove it. Or maybe I'm just trying to find, do they have amoeba or some organisms? How can I find what organisms? Oh, here's the organisms. There's a taxonomy. All right. So we're cataloging the search term words so that you should be able to find things really quick. Maybe you're like interested in orthologs. Here we go. Here's orthology, so you can see gene conservation with other organisms. And then you can follow them up, and it will take you to a strategy, a strategic mining of our data. Okay, and we're going to do an exercise on that today too. So I'm not going to take time from your exercises where you get to explore to open up any of these, but I can always clear my searches. I can get more information on how to use them, and I can expand all. Just to give you an idea of how many kinds of searches are here, there's a lot of them, okay? So, and then I can just collapse them all and clean it up. The last part of the page I want to draw your attention to, following up, David had pointed this out nicely. Each of these is a hyperlink, right? So if you wanted to go to CryptoDB and you don't remember what the address is, you can just click on Cryptosporidium or Fungi or Giardia and you'll immediately be taken to that project component. And so that's really useful way to navigate around. But importantly, I want to point out the new community chat. All right, and so I have already joined. If you haven't joined it, it would ask you to join. And this is a place where you can meet a community through a tool called Gitter, which I believe I have open here. There's a number of members, and this is a place where we can each answer each other's questions, seek help, seek guidance, share things, etc. And I want to end by going back to the front page and pointing out one really important thing. In addition to contact us, which is one way to share data, I want to point out that you can download bulk data, and there's more icons here and things to do. You can submit data, and there's links that will help you and pages that will explain how and what format it has to be in to submit data to us, and also the curation and annotation efforts that go on by a portion of the VEuPathDB team to ensure that the genome sequences that you have for those organisms that have curation are supported, and we're getting ready to move towards community annotation. I thank you for your attention. I'm going to stop there.